Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject recombinant DNA technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Today's topic is cDNA libraries. In this, you will be learning about what are DNA libraries and the steps involved in the construction of the cDNA libraries and what are the different screening methods employed to select the cloned cells and the advantages and limitations of cDNA libraries and the differences between cDNA libraries and genomic libraries. Dear learners, let's study about the cDNA library in detail. The cDNA library, it represents the mRNA population present in a cell of a particular type of tissue or a particular developmental stage in an organism. This cDNA library is small as it is used to clone only the coding sequences, that is exons. For this type of library, the starting material is complementary DNA. This cDNA is the complementary sequence of mRNA. So these cDNA libraries are useful to identify the genes that are active in one cell type and inactive in other cells. For example, if you take insulin gene, it is active only in the pancreatic cell. This gene is also present in other cells, for example skin cells, but it is inactive in the skin cells. And these cDNA libraries are also used to identify the genes which are active at a particular developmental stage. For example, if you take the hormones like estrogen, testosterone, these genes, they are active only at that is at puberty stage, after the puberty stage. Before the stage, the genes may be present but they are in inactive condition. So in this way, the cDNA library, they are represent only those genes that are expressed in a particular cell type or a particular developmental stage of an organism. So to make the complementary DNA, you have to first isolate the mRNA from the cells, that is the messenger RNA from the cells. If you take the structure of the mRNA, it will be having a 5' cap. This is the modified guanosine nucleotide, and it is having this 3' and you will be having a poly A tail. That is the adenosine nucleotides, they are repetitively they are present, poly A tail, and the 5' and the 3' ends, they are containing untranslated region, and in the middle portion, this is the actual coding sequence for the protein. In this way, a typical mRNA it looks like, and this particular, that is you should also remember the Chargaff's rule, that is the nucleotide A, it always forms 2 hydrogen bonds with T, and this pairing is very specific. A pairs with T and G pairs with C. AT pairing, it will be involving 2 hydrogen bonds, whereas GC pairing, it involves 3 hydrogen bonds. So exploiting this particular feature, that is A pairing with T, the mRNA population, it can be isolated from the total RNA pool using a poly T affinity column. So you know that the cell contains 3 types of RNA, mRNA, tRNA, and this rRNA, ribosomal RNA, transfer RNA, and messenger RNA. So the point of interest is only mRNA. So if you want to isolate only the mRNA from the pool of the total RNA, so if you take a poly T affinity column, it is poly T means it is having this thymine bases, they are added in the matrix of this affinity column. Because our particular mRNA is having poly A tail, it will be binding with this poly T affinity column. So here you are seeing the same thing. So this is the cell lysate which is containing tRNAs, then mRNAs and rRNAs. So if you pass it through this affinity column, only this because this see here to the poly T basis, this only our mRNA is getting attached, and if you wash the column with the buffer, the remaining unnecessary things, that is the tRNA and the ribosomal RNA, they will be eluting out and they will be washing away, and later you can collect our molecule, that is the this mRNA which is having this poly A tail. So in this manner, by using oligo DT column chromatography, you can isolate the mRNA from the pool of total RNAs present in the cell lysate. So then the next step is you have to prepare the complementary DNA from the RNA. So this different approaches are there. So that is you will be seeing the synthesis by means of reverse transcriptase, first strand synthesis. So whatever may be the approach, you will be the first three steps, they are the common. So first from the RNA, a DNA strand is synthesized by means of the enzyme reverse transcriptase. Then you will be having DNA RNA hybrid. So the RNA part of that hybrid, it is removed by using certain enzymes or by means of alkaline hydrolysis. The RNA strand, it is removed. Then from the single stranded DNA is converted to double stranded DNA using this polymerases. So these are the three common steps seen in all of the approaches. So this is the method of production of this complementary DNA by means of homopolymer tailing method. So here you are seeing that this is our mRNA having this 5' cap and poly A tail. So when you are adding reverse transcriptase and the that is the building blocks DNTPs, this RNA is taken as the template. From that one, this single stranded DNA is synthesized. So here poly T tail, it is acting as a primer and it synthesizes the that is the chain extension will be occurring. The cDNA is complementary to that of the mRNA. So later this mRNA, it is whenever you are subjecting it with alkaline hydrolysis, the mRNA, it is removed and you will be getting that is a single stranded, because single stranded complementary DNA. So it is having the poly C tail at one end and the poly T tail at other end. So if you add this, because you are having the C this cytosine bases at one end, if you add this oligo DG this fragments, it will be acting as a primer and it helps in the synthesis of the second strand. So here the reverse transcriptase is nothing but it is acting as a RNA dependent DNA polymerase. This DNA polymerase function of that particular enzyme, it helps in the synthesis of the second strand and you will be getting a double stranded complementary DNA. So this is the picture of generation of cDNA from mRNA by Gabor-Hoffman method. So you can view this cDNA generation in the coming animation. So once you have generated the double stranded complementary DNA, the next step is addition of linkers. So the purpose of adding linkers is it converts blunt ended molecules to sticky ended, and because our double stranded DNA, it is blunt ended, when you add linkers on either side and when you cut it with the restriction enzyme, it produces the double stranded complementary DNA with 5' protruding ends or 5' sticky ends. So here the linker taken is eco-R1 linker. This eco-R1 linker is having eco-R1 restriction site. So whenever you are adding these linkers on either side and cutting with this particular eco-R1 restriction enzyme, you are getting 5' overhangs or 5' cohesive ends. So the same thing you can view in the coming animation. So up to now we have discussed about the important steps involved in the construction of cDNA libraries in detail. So they include from step 1 to step 10. So that is in the steps 10 we have also seen that the double-stranded cDNA products, they are digested with the restriction enzymes to convert them into sticky ended fragments, and in these cohesive ended double-stranded complementary DNA, it is ligated with the vector molecule. So when you combine the cDNA with the vector molecule, you will be getting a recombinant vector, and these recombinant vectors, they are transformed in the E. coli cells to generate the individual clones. So these clones, that is the population of these cells, they represent they carry the specific DNA derived from a particular RNA. So the end result is a cDNA library which is consisting of a population of many different clones, and each clone, it carries a specific complementary DNA which is derived from a single mRNA. So the same thing, all the steps, it is present in this diagram. So this is the cell lysate which is containing total RNAs, mRNA, rRNA and tRNA. So when it is subjected to this oligo-DT column chromatography, only our mRNA molecules, they are selected, and the rest of the RNA molecules, they are washed away. And so you are, after selecting our mRNA, when you are adding this oligo-DT primer in the presence of the enzyme reverse transcriptase and the building blocks, you will be able to synthesize a complementary DNA. So this is DNA RNA hybrid. So the RNA portion of this hybrid, it is removed by means of either alkaline hydrolysis or by using this RNase H enzyme, ribonuclease enzyme. You are degrading the RNA portion, and the next step is conversion of the single-stranded DNA to double-stranded DNA. After that one, you are adding linkers on either side of this complementary DNA to convert the blunt-ended fragment to sticky-ended fragment. Later, this double-stranded complementary DNA is added to this vector, and you will be getting a recombinant vector. These recombinant vectors are used for transformation in the host cell. This particular population, they represent they will be representing the cDNA of this particular mRNA. So this is another diagram of a cDNA library. Here you are seeing 3 complementary DNA molecules, and when these 3 cDNA are introduced in 3 separate host cells by using vectors, so each bacterial cell will be multiplying and producing colonies, and each colony, it contains clones of these bacterial cell, and each clone, it contains a specific complementary DNA, specific cDNA, which is complementary to the single mRNA. So here you are having a red color cDNA, and it represents the this red color mRNA, whereas this yellow color cDNA, it represents it is complementary to the yellow color mRNA. So in this manner, each clone, it represents the specific cDNA which is complementary to the single mRNA from which it is derived. Coming to the next slide, screening of libraries. So different methods are employed to identify the recombinant host cells from the population of this non-recombinant cells. So it includes, first one is the antibiotic resistant marker. This is a selectable marker. So when you use this marker, you can easily select the transformed cells as they are antibiotic resistance, whereas non-transformed cells, they are susceptible to this antibiotic. So when you plate the transformation mixture in a medium containing antibiotic, only our transformed cells, that is the recombinant cells, will be having the ability to grow. The second method is blue-white screening. So here the cells, the recombinant and the non-recombinant cells, they appear in two colors. That is the white colonies, they represents the recombinant cells, and the blue colonies, they represents the non-transformed cells. So just by this visual appearance, you can screen the transformed cells. The third method is plaque formation. So the recombinant cells will be having the ability to form plaque formation, that is the clear zones where the particular cells are growing. They represent, this clear zones represent the recombinant cells, whereas the turbid colonies, they represent the non-recombinant cells. So detailed explanation of these screening methods, so you can go through my previous videos, and in the coming video you will see the animation of another method of screening. Moving on to the advantages of cDNA library. So these libraries are useful to identify the genes which are active in one cell type and inactive in other cells, and also they help in revealing the genes which are responsible for cell differentiation and specialization. These cDNA libraries are used to study the relative gene expression in a normal cell and in a diseased cell, how it is varying, you can study that one. And also it is used to express the eukaryotic genes in bacteria, because these cDNAs, they don't contain introns, you can easily express them in bacterial cells. It also helps in expression of a particular cell type or a particular developmental stage of an organisms can also be easily represented in a cDNA library. And coming to the limitations or the disadvantages of the cDNA library, it contains only the sequences that are present in the mature RNA, and intron and any other sequences which are altered after transcription, they are not present in this cDNA library. Also, the sequences, the regulatory sequences such as promoter, enhancers and untranslated regions, they are not present in the cDNA library, and it does not represent the entire genome of the organism, is not represented in this cDNA library. So these are some of the limitations of cDNA library. Coming to the differences between a genomic and a cDNA library. So this genomic library is large and you can prepare directly from the genomic DNA of the cell, whereas cDNA library is small because it represents only the translated region. The genomic library represents the entire genome of the organism, that is both exons and introns. cDNA library represents only the expressed gene sequences, that is the translated regions, that is exon sequences only. The restriction endonucleases and ligases are the most important enzymes which are required for this construction,
whereas the important enzyme for this cDNA library is reverse transcriptase. And this genomic library represents both the DNA of eukaryotic as well as prokaryotic organisms, but cDNA library it represents only the DNA of eukaryotes. The next difference is the eukaryotic genes from genomic library could not be expressed in prokaryotic system due to the absence of splicing mechanism, whereas these genes can be expressed in cDNA libraries because these genes they lack the introns, because you are taking the complementary DNA and that one is lacking this introns, so the eukaryotic genes can be easily expressed in a cDNA library. Then the vectors like cosmids, BARC, YARC are preferred because they are able to accommodate large sized DNA insert. Because the eukaryotic genome is large, you will be preferring the vectors which can incorporate large sized vectors inserts. This here in case of the cDNA library, you will mostly preferred vectors are these plasmids, phasmids and phases because they can accommodate small size DNA insert. So at the end, let's rewind what we have covered in the previous slides. So we have learned that the cDNA library is the collection of cloned DNA fragments which represents all the coding sequences of an eukaryotic genome, and how you have screened the cDNA libraries by using different selectable markers, blue-white screening and plaque formation, and you have learned even the advantages of this particular cDNA library, how they are used to know the structure and function of the coding sequences, and in the limitations you have learned that these cDNA libraries they don't represent the entire genome, and they can be easily expressed in the prokaryotic system due to the lack of introns. So these are the main points. With this I end the session.
Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.